Our final discussion in this chapter will focus on some technology related to the DNA information that we have discussed during the course of our chapter. The first thing we want to talk about here are stem cells. And so stem cells are a very important and very controversial topic in the world of biology. And stem cells are going to be related to some of the topics that we talk about in this little mini discussion. So stem cells come in two forms, and we need to be careful at this point to distinguish between those two forms. We have what are referred to as embryonic stem cells, and then we have what we refer to as adult stem cells. Embryonic stem cells are the primary focus of the discussion that we have on the screen. Embryonic stem cells are cells that are present during the first brief period of development in humans or any other organism. And so these cells are completely undifferentiated. You realize that based on the knowledge you've gained during the course of our class, the key in development is DNA expression. And this is true because every single cell in an organism's body has exactly the same DNA, but certain DNA is expressed in one cell that makes that cell, for instance, a nervous system cell, and other DNA will be expressed in a different cell, and that will make that cell, for instance, a liver cell. And so stem cells are very powerful because stem cells have the ability to become any type of cell, or any type of tissue, or any organ for that matter. In fact, they're so powerful that, as we talked about earlier in our class, if those stem cells split into two for some reason early in development, then two completely normal organisms develop instead of one, and that's how monozygotic twins come about. And so stem cells are very powerful in what can be done with them in terms of research in the treatment of disease. Stem cells have been used to treat diseases such as Alzheimer's, multiple sclerosis, heart disease, and stem cells can even be used to grow specific organs, areas of skin, portions of the heart, or even replace cartilage. Now, this is extremely controversial, though, and the reason for this is that currently, in order to use stem cells in a therapeutic sense, an embryo has to be destroyed in order to do that. And so, of course, that brings in it's an extremely controversial, and many people would agree that that's actually destroying a human life. And so there are many different aspects of ethics that circle around that. But I do want to make it clear, although it certainly is controversial, and I personally have issues with it, but I do want to make it clear that there are many positive things that can come about from stem cells, the actual use of stem cells in treatment. And so the good news here is this, that as our knowledge of DNA advances, it seems more and more likely that we're going to be able to take adult stem cells and turn those into embryonic stem cells. Now, let's define adult stem cells. Adult stem cells are cells in your body that are differentiated, but they never completely go to their final stage. And so here's an example. So you have skin cells. The basal layer of your epidermis is referred to as the stratum basale. The stratum basale divides continually throughout the course of your life, producing new cells. And so your skin is constantly being regenerated, and the cells are falling off. Dead skin cells are constantly falling off from the outside of your epidermis. And so that's an example of an adult stem cell. And so adult stem cells are really not very controversial for almost everyone. Few people would object to removing a small number of skin cells or bone marrow cells or other types of adult stem cells from an individual, as long as they consented to that. And so the hope is that we're going to be able to take adult stem cells and modify them genetically and make them into embryonic stem cells. A recent study published by scientists in Japan indicates that they, or lays out how they modified 7 different genes in adult stem cells and made them very much like embryonic stem cells. And so stem cells are a very controversial and very important topic, and hopefully the technology will advance in the coming years so that we can avoid this really controversial ethical issue regarding embryonic stem cells. We want to talk now briefly about the Human Genome Project. The Human Genome Project was the project that was undertaken to sequence the entire human genome. And so this was a big deal when it was first undertaken. And it's been completed now for a number of years. And so at this point, the entire human genome is known. Every single base that's found in our DNA is known. Now, of course, you realize that the DNA in any two people, aside from identical twins, is not identical. We've talked about DNA fingerprints and various topics. But it is true that DNA in any two humans is fairly similar. And so we're aware of the location of all of these genes, all the 23,000 genes that make up a human. What we're not aware of is what those genes do. And so what we're saying here is that we know, for instance, where the gene for, let's use the example that I talked about earlier, the melanocortin receptor. We know where that's located, but we don't know, of course, your sequence for a melanocortin receptor, unless you've had yours sequenced for some reason. But we do know where it's located in human DNA. For instance, we know it's located on chromosome 17 at a particular loci or a particular location on that chromosome. And so the Human Genome Project then was completed by two individuals, Craig Venter, shown here on the front of Time magazine on the left, and Francis Collins, shown here on Time magazine on the right there. And so Craig Venter was in charge of the private enterprise that was working on this project. It was actually his own company, Celera Genomics. And on the right, Francis Collins was in charge of the NIH, National Institutes of Health, funded, in other words, the government-funded effort to sequence the entire human genome. It's an interesting story. They were basically in competition to see who could finish first. Most people would agree that Craig Venter finished a little bit before Francis Collins did, but they both share credit for this accomplishment. Now, I will point out, I need to update this slide. So this slide says that humans have 20,500 genes, and so we've adjusted that a little bit to 23,000 at this point. Most of these genes code for proteins, and interestingly, most of the average protein coding gene in humans is an intron. And so we have, within the actual gene, we have two regions. We have introns and exons. Exons are the regions of DNA that actually code for bases, and then introns, fascinatingly, are these regions within a gene that don't code for anything at all. And so what we're saying then is that within a particular gene, there are little mini segments of DNA that are eventually going to be cut out. This is not something we talk about in great detail in our class, but this is a process known as splicing. And so if you think back to what you've learned in this chapter, you know that we go from DNA to RNA. And so once you've made the RNA, interestingly, in that RNA you go through this process of splicing, and you cut out the sections of RNA that are not going to be part of the protein. And this is very important in protein construction, and it's also very interesting because splicing can occur in different orders, and so alternative splicing can allow us to make as many as 1,000 genes from just one particular RNA sequence. So imagine that you cut out sections of RNA, and then you glue the pieces that are left together back into different orders. If you glue it together in one order, you come up with protein A. If you glue it together in a different order, you come up with protein B. And so what I've talked about before is that much of the human genome was formerly described as junk. In other words, what I've told you before is that all of those 23,000 genes are found within about 2% of our DNA. The other 98% of our DNA doesn't code for proteins, but much of it does have some regulatory role. We don't really go into that role in this particular class, but it does seem to be doing something, and as time goes on, we understand more and more as a result of research about what that other 98% of the DNA is actually doing. For instance, on the screen we have a statement that says RNA molecules can have a regulatory effect in cells, and so we now understand things like silencing RNA, which I'm not going to attempt to talk about at this time, but it's an interesting aspect of the regulation of our DNA. And so there's portions of our DNA code for RNA molecules that actually regulate which DNA is expressed and which DNA is not expressed. We want to now briefly talk about gene therapy and so-called genetically modified humans. And so no one is suggesting that it would be a good idea to take humans and genetically modify them and make them, give them a certain color of hair or make them glow like we do with fish or anything like that. But what we are attempting to do with genetic modification in humans is to cure diseases. And so there's two approaches to accomplish this. One is germline therapy and one is somatic cell gene therapy. Germline gene therapy is what I should have said just a moment ago. So two types of gene therapy, germline and somatic cell. Now, germline gene therapy would require this. It would require that healthy DNA be inserted into an embryo when it's in the very early stage and just consists of stem cells. So in other words, what I'm saying very clearly, I hope, is this. You realize that cystic fibrosis is caused by one particular mutation in just one protein. If you could fix that protein, then people that have cystic fibrosis would be cured 100%. They'd be completely normal. So, if you could take an embryo in the very early stages of development and inject the healthy gene into that embryo, fascinatingly, if you just injected the DNA, then that DNA, much of it would be taken up into those cells, and that embryo would be completely healthy. At least that we've demonstrated that we can do that in animals. That's not something that's been attempted in humans, nor is it likely that it will be attempted in the near future, because it's still a pretty rough process. There's a lot of potential to make mistakes there. But that would cure cystic fibrosis. It's also, you have to also realize that no one's going to realize that the child, the embryo, has cystic fibrosis at that very early stage, and how would you do that inside the mother? There's all sorts of complicating factors. But what is being attempted is something called somatic cell gene therapy. And so you realize that somatic cells are non-reproductive cells that are found in the body. And somatic cell gene therapy involves the insertion of a healthy gene into cells that are defective, and that gene then ideally becomes expressed, and so a healthy protein is expressed in those cells. For instance, today we're working on somatic cell gene therapy to cure cystic fibrosis, and it's even gotten to the human testing stage. And so people that have cystic fibrosis take an aerosol spray that contains the healthy gene for the protein that's a chloride ion transporter that they are missing. And so that spray goes down to their lungs, that gene makes its way into their lung cells, and at least there's some indication that that gene can actually be expressed in their lung cells. And if it was fully expressed, then they would be cured of cystic fibrosis, at least as far as the effects in the lungs. It does have some effects in other parts of the body, but primarily it does affect the lungs. And so it would be tremendous for those people if that was could be made 100% effective. And you may ask, well, how does DNA get into an adult cell? And the answer, interestingly, is a virus. We have not talked about viruses in our class up to this point, but viruses actually cut DNA and insert themselves into DNA. It's perhaps disturbing to talk about, but both types of herpes viruses, for instance, the STD and the one that causes cold sores, and those are not the same, simplex 1 and simplex 2, but they cut human DNA, they spread apart your DNA, and they put themselves into your DNA, becoming part of your DNA. And that'll be with you for the rest of your life. A certain percentage, I do not remember exactly what percentage it is, but somewhere between 10 to 20%, I think, of the human population has the herpes virus that causes cold sores. And not particularly your fault, but you were exposed to someone, if you have that during the course of your life, who had that herpes virus. They transmitted it to you, and you'll have that for the rest of your life. And so the idea here is that we take a harmless virus, we attach the healthy gene to it, we put it in the spray, give that to the person, and hopefully the virus delivers that gene to the appropriate site in their lungs. And there's some indication that we've been successful with that. Excuse me, research is being done on that particular approach at this time. Alright, and so I got a little bit ahead of the notes. Germline gene therapy involves embryonic treatment, and so that would we talked about the potential complications regarding that approach, somatic cell gene therapy, involves addressing only particular cells and cells in an adult human. This is a treatment that's been used not only for cystic fibrosis but for severe combined immunodeficiency, which is a defect that involves white blood cells. And so here, this approach is being employed. Immune system cells are being removed from the patient. White blood cells are being removed. The cells, according to here, this is a white blood cell. The cell is being infected with a virus. Maybe you didn't realize this is what viruses look like, but this is a common form for a virus. And the virus inside it has the healthy allele, the gene that's missing in this little girl's white blood cells. And so the virus infects the white blood cell and becomes part of the DNA of this little girl in her white blood cell. The white blood cell is placed back into her, and hopefully that cures her of this particular disorder, as the disorder occurs because, of course, she lacks the gene in her white blood cells. And there's been a lot of promise regarding this approach in this particular disease. So in a nutshell, that's gene therapy, and the two options for how gene therapy can be approached, only one of which is currently being employed at this time. And really, it's unlikely that embryonic germline gene therapy will be applied to humans anywhere in the near future. We want to spend just the last few moments of our discussion here talking about cloning. And so cloning is also a controversial topic, but it's an interesting topic. And so cloning is has been widely conducted at this point. It's been done with cattle, goats, mice, cats, sheep, pigs, mules, some other things as well, I believe, that are probably not listed there. And we'll talk about how cloning works here in just a moment, but cloning really is becoming fairly mainstream. There is a company today that can clone your pets, specifically your cats. I'm not quite sure if it's available for dogs or not. You'd have to look into that. But if you have a cat that you really like, then you should take a hair sample from the cat. Don't clip the hair off, but pull the hair out very gently by the roots. Now, I don't grab a whole fistful, but just get a couple hairs, pull them out, put it in a little Ziploc bag, label it, and put it in your freezer. If your cat runs away, then you can mail this little bag that has your cat's DNA in it. The DNA will be found in the roots of the hair. Interestingly, the hair itself, those cells are dead, so it doesn't have DNA, but the roots do. And so you can mail this to this company along with $25,000, and they will clone your cat and send you a brand new cat that's a clone of the cat that you lost. And so cloning is relatively, now of course that's a lot of money for a cat, I realize, but it's still, it can be done. It's not a rip-off, it's something that's completely legitimate. Of course, you may disagree with whether cloning is legitimate, but it really is something that is done. And so, how do you clone them? Let's spend a moment talking about how you actually produce a clone. And here's how you produce a clone. So Dolly was the very first clone produced, and this is how you clone. So here is the donor of Dolly. I don't know exactly what term you could use besides donor. So we took a sheep and we removed a cell from the udder of this particular sheep. It could have been any cell on the sheep's body, but we happened to use an udder cell. We took that cell, and let's go down here next. We took a completely different sheep, a female sheep, and we removed an egg from that sheep. We got rid of the nucleus, as indicated by the X here, of that egg. And so here we have an egg that doesn't have a nucleus in it. And so we take our nucleus from the mammary cell and we add that nucleus to our denucleated egg from the egg donor. And so here we have an egg, an interesting situation, an egg that's not haploid but it's diploid. It has all the DNA necessary to form a new organism. And remember, this nucleus just came from one cell, any old cell in this sheep. We then treat this cell, we shock it with some very, very minute electricity, which we figured out tricks the cell, and it makes the cell think it's been fertilized by sperm, and so the cell starts to divide. And so at this point, we have an embryo that is basically normal. Now there may be some complications regarding the age of DNA and things that we don't entirely understand at this point, but it's safe to say that this embryo behaves normally. We take this embryo, we implant it back into a mother. It could be the mother here, or it could be the female here, or it could be a completely different female. That's relatively commonly done in agriculture today, and also that's what in vitro fertilization is in humans, as you likely realize. And so this embryo then matures, and the baby is born. And so here is your clone, your baby that is a clone of the cell donor, the nucleus donor back here. And so that's cloning. Cloning is certainly an interesting topic. It's something that's widely done. It's done in research circles to produce animals that are genetically identical. It's done when we get an animal that we have genetically modified that produces something very valuable, like our earlier example where we had the human growth hormone that was produced by the organism. And it's done for other things as well. One of the most interesting topics in the news right now regarding cloning is the attempt to clone a woolly mammoth. And so woolly mammoths have been extinct for thousands of years, but they, unlike dinosaurs, still exist in some places in a frozen state. And so right now there are scientists that are working on a project to clone a woolly mammoth. They have a woolly mammoth that's in an ice cave actually I'm not quite sure where it is. I'm not sure that the location has even been revealed. But the DNA from that mammoth, they're attempting to take a nucleus and use it in an elephant egg, and then of course implant the fertilized embryo back in, not the fertilized embryo, but the dividing embryo back into an elephant. The problem has been that the DNA, since it's several thousand years old, is somewhat degraded. But the hope now is that they can patch together the mammoth DNA with some elephant DNA and have something that's basically a mammoth with a little bit of elephant DNA mixed in, hopefully in areas that are not don't affect the phenotype. So that's cloning, something that's somewhat controversial, perhaps even among animals, but it's something that's widely done today and has a useful application in science.